Hi, we would first like to thank the meeting organizers for providing us with this opportunity to share our work with the scientific community. We are senior scientists in Amgen working on developing microphysiological systems and analytical methods for these models. And our presentation today will focus on the application of microphysiological systems in biopharmaceutics, as well as recent advancements we've made in analyzing these models. So let me move forward. So an MPS is a complex multicellular 3D in vitro system. And unlike traditional cell cultures, the MPS is designed to replicate the intricate environment of human tissues and organs. And these systems possess functional features that closely mimic the physiological and pathophysiological conditions of human tissues. And by creating a microenvironment that mirrors the natural conditions of human organs, MPS allows for the study of how cells behave in real-life scenarios. This advanced approach helps in understanding these disease mechanisms, testing drug responses, and developing new treatments with greater accuracy. So, in biomedical research, the limitations of animal models due to their different physiology and inconsistent cellular responses highlight the need for more accurate and convenient alternatives. Human-based models, such as the MPS models, offer a solution by closely mimicking human tissues and organs. And these advanced models establish a more precise representation of human physiological conditions, improving the reliability of research findings and potentially reducing the reliance of animal models. And moreover, the FDA Modernization Act, first announced in early 2023, started supporting cell-based in vitro models for drug testings. Thus, the biopharma industry has focused on developing and utilizing MPS models for testing candidate molecules. So then what is the difference between the conventional in vitro models versus MPS models? The 2D in vitro models, featuring cell monolayers cultured on well plates or transwell systems, offers a simple and cost-effective approach for rapidly assessing cellular responses, such as cytotoxicity and metabolism to various compounds. While it efficiently reveals biological trends and is valuable for initial testing, it actually falls short in replicating in the complex 3D microenvironment found in vivo and limiting its ability to fully mimic the intricate interactions and conditions of human tissues. So in contrast, the 3D MPS closely replicates the structure and function of in vivo tissues. And this advanced system enhances our ability to predict and understand how molecules interact within physiological context, offering a more valuable alternative to traditional animal models. Although it demands more effort to maintenance due to its complexity, its ability to mimic human physiology makes it an essential tool for more accurate and relevant biomedical research. So the key elements of MPS include carefully chosen cell sources and microenvironments to accurately replicate human tissue conditions. And this involves selecting cell-specific ECM and using various cell types such as iPSCs, primary cells, or patient-derived cells. And signaling factors are precisely controlled through gene editing, growth factor supplementation, and flow conditions. To achieve a true recapitulation of tissue structure and functionality, MPS leverages advanced computational design along with a sophisticated functional readout and analysis methods, ensuring a comprehensive and realistic model for studying human biology. So the final analyses and readouts from MPS models go beyond just molecular biology-based assessments. They also include quantifying biological and morphological data through imaging. For instance, we can measure molecule uptake by observing changes in fluorescent intensity or assess muscle tissue force generation by measuring tissue displacement. These results help compare healthy and diseased tissue states, offering valuable insights for designing therapeutics. In the remainder of our presentation, we will focus specifically on the microvascular network, or MVN, model that Amgen is currently using. In the biopharmaceutical field, the effectiveness of biologics depends largely on their ability to reach their target, with the vascular endothelium serving as the primary barrier. And traditional endothelial models often show excessively high permeabilities due to excessive paracellular transport and the absence of glycocalyx. In contrast, the MVN models offer an in vivo-like microenvironment and provide permeability values that are comparable to those of the in vivo models. So the MVN models are highly versatile for various in vitro pharmacokinetics and pharmacodynamics applications. They can be used to study drug-drug interaction, vascular permeability, and target access. Additionally, they provide insights into diffusivity, tissue retention, catabolism, toxicity, and other physiological parameters. Furthermore, MVN models have the potential to be used for generating disease models. So to create our MVN model, the endothelial cells and stromal cells are combined in a fibrinogen gel, and they're injected into a confined chamber within a microfluidic device. So over the course of 7 days, the cells will self-assemble into 3D microvascular network, forming both endothelial and extracellular tissue spaces. The endothelial vasculature is supported by the stromal cells within the tissue space, and the endothelial walls include glycocalyx content. From the MVN model, we are able to collect different data sets. We can collect the supernatant from the devices to perform Luminex or ELISA, and we are also able to retrieve tissue samples from the devices to perform various molecular biology-based analyses, such as qRT-PCR, protein analyses, and sequencing. Also, one key readout is to obtain the biological and morphological information from image-based methods by measuring diffusivity, immolar fractions, and the endothelial permeability of target proteins. All of these measurements are combined to evaluate the properties of different molecules and to study the biological mechanism that are happening in vivo. And now let me hand over the microphone to my colleague Devin to dive a little bit deeper into the details of our MVN imaging assay and associated analysis workflow.
Thank you, Claire. As Claire indicated on the previous slide, fluorescent microscopy is among the approaches that can be applied to extract a wealth of information from MVN models. Fluorescent imaging allows us to readily visualize and probe features of the microvasculature that cannot be captured by other methods. Our current work aims to use imaging to directly quantify the movement of various biologically relevant molecules as a result of measuring fluorescent intensity changes over time within our MVN device. To that end, our approach leverages confocal microscopy. The key strengths of this imaging modality for our assay include features for speed, a controllable depth of field, and ease of use for imaging thick samples tens to hundreds of microns thick, as is the case with our MVN model. So, with everything presented thus far, let me now go ahead and provide details on our experimental setup for an imaging assay. A single MVN microfluidic device is placed on the microscope and image acquisition settings are defined. The device is then scanned to identify and select acceptable regions for imaging. Next, the media channels in the device are perfused with the fluorescent proteins. This is immediately followed by time-lapse image capture. Imaging typically includes all fluorescent channels, 3 fields of view with a defined Z-stack size, and a total time of about 10 to 12 minutes. All of this has been optimized to balance image resolution with a sufficiently fast enough time resolution to capture details in the MVN structures and changes in fluorescence. To give the optimization a bit more detail, here's a table summarizing experimentation on the imaging times required to effectively measure permeability. Importantly, the imaging time can be adapted to a range of molecules according to their molecular weights. Molecules as large as antibodies, like IgG-Fab, and monoclonal antibodies were tested, and molecules as small as fluorescent dyes, like FITC, and compounds like paclitaxel were also tested. All of these have different permeabilities, and thus the required imaging time is a function of their molecular weight. Upon completion of having imaged multiple regions of interest from multiple MVN devices, our automated image analysis routine can then be performed. Before I jump into the details of our analysis, let me provide a brief overview on the state of the field for MVN image analysis. A number of image processing tools are readily available to aid in the quantification of fluorescent images of microvessel architecture. Tools like the examples shown on the slide offer tunable input parameters and features for customized detection and segmentation. However, these methods face some limitations as they may require manual selection of areas from the images for the analysis, rely on multiple image processing steps to segment the structures, and/or do not incorporate automated calculations or output of desired metrics. Moreover, a significant amount of time and effort could be spent tuning input parameters and still come up short in providing the level of specificity needed to capture the variation in structures observed in an MVM image, variations such as shape, size, and branching. Deep learning methods, on the other hand, offer a highly trainable and adaptable method for performing microvessel segmentation. The emergence of deep learning models has completely transformed image segmentation for MPS and MVNs models. In particular, deep learning image segmentation removes the tedious steps of tuning input parameters and enables the recognition of image features based on fluorescent signals, vessel morphology, texture, and overall structure. A powerful method can take large datasets of raw or processed images, learn the key features in the images, and ultimately supply improved accuracy and fidelity in the detection of microvessels. Our current approach aims to leverage the strengths from deep learning and more using an automated analysis that does not require manual image area selections or manual collection of output metrics. A trained deep learning model provides effective binarization of the 3D surface of the microvasculature for desired measurements. The main steps to our custom analysis are listed here. First, image stacks are read indirectly from the microscope. The specific segmentation on the MVN images applies a 3D UNet model. Details on the deep learning model in just a moment. From the binarized images, the necessary metrics are extracted to then calculate permeability. These metrics include the intensity values from inside and outside the vasculature, the total volume surrounding the vasculature, and the surface area of all segmented vessels in an image. And the final step in the analysis is a complete summary file that is automatically saved to provide for downstream quality control, visualization, and evaluation of results. The full analysis process takes just only minutes for an average single data set of about 15 images or 5 MVN devices. Conveniently, a simple user interface has been designed to house all the codes and ultimately allow for minimal user input of just the sample details and file selection in order to kick off the analysis. As mentioned, we have a UNet model trained to perform the 3D segmentation. The legendary UNet model architecture has been widely applied to many biological image segmentation tasks with great success. To prepare our model, ground truth images were annotated using the open source software tool Elastic. You can see that from quick tracings or fast scribbles within the images to indicate regions inside and outside the vessel structures, we can thus readily prepare some simple ground truth segmentation for model training. The model itself was trained on data from multiple experiments to include over 300 small Z-stack image patches. On a trained model, the segmentation quality was inspected and analyzed. Comparison of the ground truth images with the predicted unit segmentation images reveals strong agreement and an accurate segmentation model. This process was repeated several times to help the UNet model improve and to ensure reproducibility. Here we are on my favorite slide with two nice examples, one on each row of what we typically see from the 3D segmentation. The middle panels illustrate stepping through each Z-plane to view the Z-stack image and segmentation masks. On the far right is the very mesmerizing final rendering of the 3D segmented surface. A key takeaway here is to see just how well the trained UNet model can overcome noise in the sample and adapt to the significant variation in the MVN morphology. Such levels of heterogeneity are not only found between MVN devices, but can sometimes even appear within a single MVN device. Lastly, provide a very quick highlight on one result. Here we have performed our MVN assay on two different molecular weight dextrins. And nicely, as we would expect, the small molecular weight dextrin shows increased permeability. If you're interested to learn details about the specific protein studied with our MVN assay, I encourage you to visit our colleague's presentation on the applications of cellular assays for understanding drug disposition of protein therapeutics. To wrap up our presentation, we introduced our MVN model in a microfluidic device that closely mimics in vivo microvasculature. We've optimized our MVN protocol for preparing and analyzing this model for routine use and robust results. A trained 3D UNet deep learning model has allowed us to achieve accurate microvessel segmentation. Importantly, accurate segmentation is essential for accurate measurements on molecule permeability and for our ultimate goal of improving molecule characterization and design for Amgen. Our automated MVN analysis method has begun yielding insights beyond the capabilities of traditional 2D approaches, significantly broadening the scope of possible studies with MVNs. Our MVN system is not limited to measuring permeability and could be applied to understanding cellular processes in healthy and disease states, as well as modeling fluid flow and other aspects of mechanobiology. Moving forward, we plan to utilize this model and assay to explore the behavior of various candidate molecules within the MVN and to study biological processes that occur in vivo post-injection. Thank you to our respective department teams at Amgen, and thank you for your interest in our exciting research.